Good morning, everyone. Welcome to the Assay Guidance Manual and CAT's Assay Guidance Manual Preclinical Translational Science webinar series. For those of you who don't know me, I'm Serene Markosian. I lead the Assay Guidance Manual program here at NCATS NIH. And today we have a super special guest speaker. So super excited to have Dr. Michelle Arkin with us. Michelle is my previous mentor, so I'm absolutely thrilled to have her today. She is actually also a distinguished chemical biologist, professor and chair of pharmaceutical chemistry, and executive director of the Small Molecule Discovery Center at University of California, San Francisco. I'm super excited to have her as well, a member of the Assay Guidance Manual editorial board, and of course, several other chemical biology journals. Her research focuses on developing methods and molecules that target currently undruggable proteins, including protein-protein interactions and dynamic or intrinsically disordered proteins. She is also a co-founder and director of Ambagon Therapeutics, co-founder of Elgia Therapeutics, and advisor to several other biotechs. Prior to UCSF, Michelle was associate director of cell biology at Sunesis Pharmaceuticals, where she helped discover inhibitors of protein-protein interactions for IL-2 and IL-2R and LFA1 ICAM, which is lifitegrast, marketed by Novartis. And if I misspelled any of that, Michelle, I apologize.
No, you did great.
So she earned her PhD in chemistry at Caltech with Jackie Barton, and held a Damon Runyon postdoctoral fellowship at Genentech in the lab of Jim Wells. She was recently named a 2024 Arthur C. Cope Scholar by the American Chemical Society. We're super excited to have you here, and on behalf of all of my colleagues here at NCATS, Michelle, we welcome you, and we're thrilled to have you here and look forward to your talk. Michelle's talk is entitled Cysteine-Directed Molecules as Drug Leads and Probes of Undruggable Proteins. The floor is yours, Michelle. We're excited.
Thank you so much. Thank you, everybody, for being here and for Zooming in. So I'll start. I'm going to talk about one project. I'm always tempted to talk about a whole bunch of different things because there are a lot of exciting projects going on in the lab. And I'll talk about one project that was funded by the Ono Pharma Foundation and now funded by NIH. But this group of people has so many exciting things that we could be talking about. And so maybe next time I'm invited, we'll talk about exciting work going on from Andy Ambrose in Caspase-6 or Chad Altobelli on P97 or some of these other great projects. So I just want to thank ahead of time this fantastic team. And the project that I'm going to talk about is based on 1433 and molecular glue design. And I recently gave a talk at the Academic Drug Discovery Consortium meeting, and they wanted something more about how teams get together. And so I'm using some of those slides because I thought they wound up being kind of fun to talk about the people-centric way of doing science and of starting collaborations. And collaborations are so important to biomedical science and so important to my own career that it's fun to focus on it that way. So with that in mind, our story begins in Berlin in 2012, where I saw this amazing talk by Christian Ottman, who's the guru of 1433. So I'll describe what he talked about. So 1433 is this protein. Let me use this instead. Not the highlighter, goodness. 1433 is this dimeric alpha helical protein. So here's the dimer interface, this blue protein. It binds to phosphorylated proteins. And in plants, it's known that binding to this ion channel and ATPase protein leads to ion channel opening and increases the ion channel at the surface. So it has an agonist function. And there's a natural product, a fungal natural product that kills the plant. And it does this by binding to 1433. Here's the compound shown in orange. Binds to 1433, stabilizes its interaction with its ion channel, which increases trafficking, increases ion channel opening, and kills the plant, which then the fungus eats. So during his PhD, he saw this crystal structure and characterized how this natural product worked. And he saw right away that this could be used for human biomedical applications. And if we zoom in on how this protein-protein stabilizer works, this natural product stabilizer, here's again the binding groove in 1433 for this phosphorylated peptide. You see this green peptide coming from the ion channel. And fusacock in this natural product binds at the end of the binding site and acts like a cap or a wad of gum that glues the ion channel to 1433. And so this is what we wanted to try to do together. So 1433, as I mentioned, could have a lot of biomedical applications. That's because it's the central effector of serine and threonine phosphorylation in the cell. So if you work on kinases, you work on 1433, because one of the key things that, one of the key reasons that a protein is phosphorylated at serine and threonine is to make it a client for 1433, and then 1433 acts like a shuttle or a taxi, can move proteins in and out of the nucleus up to the cell membrane, as it does with this ion channel. It can also break protein-protein interactions by blocking them. It can stabilize protein-protein interactions by having two proteins bind in this dimeric site. So the key thing to understand about 1433, it can do all these different things because its function is really encoded in the protein that's phosphorylated. So we'll see this a couple of times where the phosphorylation site dictates what 1433 is going to do to that protein. If it covers a nuclear localization signal, or it binds at a dimer interface, or it allosterically blocks a dimer interface, you can see where this is going. So because of that, because the activity is encoded in the protein itself, 1433 can act as an activator or an inhibitor. And as it happens, there are many undrugged proteins, especially intrinsically disordered proteins, that are clients for 1433. And there are also, as you think about it, there are high concentrations of 7 highly homologous isoforms. So to use the taxi analogy again, if what we do is try to stabilize one protein-protein interaction, you may be concerned that there aren't enough 1433s around to do the rest of the business of the cell, but there are. It's like when you step out at JFK and there's a long line of taxi cabs, okay, of different brands of taxi cabs, but they all do more or less the same thing. That's how we can think about 1433. There's a lot of it around. There's always enough for proteins to be stabilized and still have plenty more to do other functions of 1433. So what I talked about at this Berlin meeting in 2012 was about what makes protein-protein interactions so-called undruggable targets. And the basic thing that I talked about at the time was this flatness problem. So if you look at the protein-protein interface, one protein is shown in gray, the other protein partner is shown in blue, and the structural interface, so the residues from one side that are covered by the other protein, are shown in pink. So that's a very large surface area. It's a fairly flat surface area. And so the thought had been that that this protein, if you wanted to bind to this protein, it was like trying to bind to a cliff face. It was largely flat, it was large, and to find a molecule that would bind to that surface would be akin to finding this person hanging on the side of this cliff with just very small subsites, small holes in the protein interface that a molecule could bind into, like this woman's fingers and toes, and the rest of the molecule goes to making those interactions possible, so to scaffolding those interactions with each other. Like most of her body goes to scaffolding the interactions with the wall, with the rock face. So we would say in chemistry that she has a low ligand deficiency because most of her is scaffolding the interactions and a very small amount of her is actually interacting with the wall, so making contacts with the wall. So the analogy for drugs then is that the molecules might be very large because they're trying to access many small pockets. And the way that we would approach this kind of problem is using a fragment-based discovery approach. So instead of making a molecule that has a predefined shape and hoping that it can make all of these little interactions across this large surface area, Instead, we discover the pieces one at a time. Again, to use the analogy of the rock climber climbs up the wall, a hand and a hand and a foot, rather than jumping onto the wall in a predefined space. So this allows us to find these little pieces, so each binding subsite, optimize that, and then link them in another step. And I think there was another presentation in this webinar series by Steve Fessick, who's really the godfather of this field, using SAR by NMR to identify fragments. And then we link them in a separate step. But now all of our chemistry is pre-organized towards fragments that we know bind to the surface. And it's a question of how to get those fragments linked together, which is a harder problem than it sounds. So I was talking about this, and then Christian reminded me, I talked to him after our talks, that there's a second reason that protein-protein interactions are so-called undruggable, and that's because many protein targets are intrinsically disordered when they are not in their protein-protein complexes. So a number of proteins like MYC, YAP, and TAZ, a number of transcription factors are more intrinsically disordered. They might even be in phase-separated states. And it's very difficult to figure out how you're going to find a molecule to bind to an intrinsically disordered protein because there isn't a binding site at all. It's like trying to climb through quicksand. But the key point is something I just alluded to, that these intrinsically disordered proteins are actually form structures when they bind to other proteins. So if we can't touch the protein itself with a small molecule, perhaps the structure that's formed once these protein-protein interactions are formed, they're now in a stable conformation that might have pockets that small molecules can bind to. And if we can find molecules that bind and stabilize that structure, we may be able to modulate the biology in the proper direction using that. So now instead of looking for inhibitors, we're looking for stabilizers. Functionally, physically, they're stabilizers, and whether they inhibit or activate this particular system depends on the nature of that system. So we decided to work on this together because 1433 is a great model system, having an intrinsically disordered protein partner, and 1433 itself is highly structured. It's a really good model system to try to develop molecular glue stabilizers. It's also from a chemical biology perspective, it's really exciting to think about stabilizing protein-protein interactions. So there are a couple of reasons for this. One, natural products like rapamycin and fusacoccin function in this way. So this is a mechanism that nature has used to manipulate protein-protein interactions. Second, looking at these two targets tells you a lot about what we might be able to do with chemical biology if we know how systematically to stabilize protein complexes. So rapamycin, the orange molecule here, binds to a protein, creates a new surface that then recruits a second protein. Now, these proteins don't normally interact with each other. So this we would call a synthetic interaction or a neomorphic interaction. And secondly, the function of this, making this protein complex, is to inhibit the function of the protein that's being bound, of mTOR. Fusacoccin, on the other hand, we already showed this. This molecule stabilizes a protein-protein interaction that is natively formed. And by increasing the binding, it activates this protein. It increases ion channel trafficking for this protein. So with chemical biology, we, in principle, could work on neomorphic or synthetic interactions or stabilizing native interactions. And depending on what targets we choose, we may be able to inhibit biology or activate biology. And there are a couple of examples of small molecules that also, synthetic molecules that act as molecular glues, and they already show this range of biology. So the lenalidomides or the imides, a very famous molecular glue, probably you guys all know about it, but it binds at this site, binds to a ubiquitin ligase. At this site, recruits transcription factors that are not normally recruited by cereblon, by this receptor, lead to ubiquitination and degradation of this target. Okay, so this, again, would be a synthetic interaction that leads to degradation, inhibition of that target. The integrated stress response inhibitor is a molecule that Peter Walter's lab developed in collaboration with our group. This compound, oddly C2 symmetric compound, binds at an interface of two large oligomers that come together, a heteropentamer. Two heteropentamers come together to make this decamer. And it's a C2 symmetric decamer. And this molecule just binds right in the middle at that interface, at that decamer, the pentamer-pentamer interface. So it stabilizes this native complex, and it makes this native complex insensitive to phosphorylation. So there's a lot of chemical biology we could develop here. The problem, if you could say, with lenalidomide and ISRIB is they were identified through phenotypic screens, and only later was it found out how they worked. So in the last several years, there's been a lot of amazing work trying to be more systematic about how we could discover molecular glues in the degrader space, and we want to bring that kind of systematic study to the non-degrader space. Here's the last reason really motivating these studies. If you look at a protein-protein interaction map like this one, this is identified through mass spectrometry. These are proteins that bind to each other. And if you look at this protein here, which happens to be 1433, it interacts with many other protein targets in many different biological networks. So epidermal growth factor receptor, so this would be growth factor receptor. This is CDK would be cell cycle, MYC transcription, DNA damage and repair, and so forth, mTOR, so autophagy. So if we knocked this out, if we wanted to understand how 1433 behaved in this cancer situation, and we knocked it out, the most specific knockout you could possibly imagine, it still would really not tell you very much about how it functions in this disease, because you would knock out all of these interactions. But if instead we could just inhibit or stabilize one interaction, so instead of a node knocking out a whole protein, an interaction edge, say between these two proteins, then we could really understand what the function was of 1433 in this, maybe the cell cycle biology. Does it stimulate cell cycle control? Does it inhibit the cell cycle? And then we would really have a better understanding of this network. So in principle, if we can stabilize a protein-protein interaction, we could really amplify this edge and understand much more about the biology or a single function of a multifunctional protein. Okay, so our goal is to systematically stabilize specific predetermined 1433 client complexes. And we're going to start with the cancer targets, and what I'll show you now. So last piece of information to understand before we go into the data is that the protein-protein interactions that we're talking about, here's 1433 now shown in white, and BRAF with MEK kinase bound to it, shown in light blue and dark blue. These proteins are difficult to express, they're difficult to work with, they need to be specifically phosphorylated at specific sites in order for us to study the protein-protein complex. And so while we can do that for biophysics and for structural biology, really for small molecule discovery and high throughput screening, it's much easier to work with a model system. And so instead we work with a peptide that's derived from that protein at that binding site. And these are a crystal structure with a dark blue peptide overlaid with the cryo-EM structure of BRAF that I just showed you in light blue, just excising that piece of the cryo-EM structure. And you can see that the peptide is a faithful representation of the protein-protein complex. So we'll be looking at protein peptide stabilization in vitro, and then when we go into cell-based assays, then we'll be looking at the full-length protein-protein interactions. Okay, so how are we going to use our knowledge of protein-protein interaction biology and inhibiting or fragment-based drug discovery to find these molecular glues? So we started with a technology that was developed at Synesis when I was there by Andrew Braisted and Dan Erlinson, called disulfide tethering. And in this approach, you have a protein with a thiol with a cysteine residue that's near a putative binding site. This can be engineered or wild-type cysteine residue, and a library of molecules that have fragments that are meant to bind to the protein, and these mixed disulfides. And ththese are just little solubilizing groups that are meant to float away. And we mix these under conditions that favor thiolate disulfide exchange, so we can readily go back and forth until we achieve thermodynamic equilibrium. And at thermodynamic equilibrium, we'll identify molecules that have non-covalent affinity, that are complementary to that binding surface, that are binding fragments, and they'll also have the correct geometry to make this disulfide bond. That's what makes this approach site-directed. Okay? We'll only see things that make this disulfide bond because we'll look at the protein now by intact mass spectrometry, and we're looking for the adduct mass shift between the unmodified protein and now the fragment modified protein. So this has quite a few advantages for doing a molecular glue discovery. One is because it's site-directed, we can choose the location. So it could be that the protein-protein interaction site is the richest site for binding, but we don't want to be at the protein-protein interaction site. We want to be right next to it or at a little pocket in the interface where we can stick a molecule to then glue them together. So we can decide where we think a good position is. And two, because it's a reversible chemistry, we can tune the reactivity by tuning the reduction potential. And this allows us to either make a very stringent screen if we want to separate high affinity things, or a very non-stringent screen if we want to saturate the binding site. And so this allows us to screen for function. This is what the fragment library looks like. There's a linker and then the fragment at the end of the linker. And this linker varies, can be short, go straight to the fragment, or it can be highly elaborated. Here we have basically an unnatural amino acid stuck in between the fragment and the disulfide. So this length and geometry is often an important aspect, important product of the screen. Okay, so our first target is going to be estrogen receptor. At estrogen receptor, 14-3-3 interactions are fairly straightforward as far as 14-3-3 biology goes. So estrogen receptor is this dark blue protein here that's bound to HSP90, so it's bound to chaperones in the absence of ligand. When estradiol enters the cell and binds to estrogen receptor, it can be released from its chaperones, and then it has two choices. It can dimerize, bind to response elements, estrogen response elements in DNA, and mediate transcription, and this can lead to breast cancer. But also when the protein is bound to estrogen, there's a change in conformation that leaves the C-terminus now available to be phosphorylated. And if the C-terminus is phosphorylated, then ER-alpha becomes a client for 14-3-3, which keeps it from binding to DNA. So it works differently from the estrogen receptor drugs that we already have, which stabilize binding to DNA. Another reason that we're interested in looking at this target, even though we have many highly potent compounds that inhibit estrogen receptor and are used in breast cancer treatment, they all bind to the ligand binding domain. And therefore, any compound or any mutation in estrogen receptor that renders a compound no longer bindable, that weakens the binding to these inhibitors, may also render estrogen receptor not bindable or not inhibitable by all of the estrogen receptor ligands. Okay? So they're all close mimics of estrogen. So if instead we target the C-terminus here, now that's phosphorylated, that binds to 14-3-3, it works by a different mechanism, may have completely different mechanisms of resistance. So you can imagine using these in combination. This is a structure of the estrogen receptor C-terminal peptide. It's phosphorylated at the penultimate residue here, and then the C-terminal residue is valine. This is the estrogen receptor peptide bound to 14-3-3. Here's fusicoccin bound, so you can see how it acts like this wad of gum that I described that really fills the rest of this potential binding groove. So we're using this as inspiration. There's a cysteine residue at the distal end of that binding site in one of the isoforms of 14-3-3. So the other isoforms won't be touched by this cysteine-driven technique. And whether that's a bug or feature will depend on the biology, but that's the sigma isoform specifically has a cysteine residue. So in this first study, we studied the cysteine residue that's wild type, but we also knocked it out or mutated it and added other cysteine residues along this helix, getting closer and closer to the binding site, because we didn't know if our fragments would prefer to be tethered close to the peptide or far away from the peptide. And then we did the disulfide tethering experiment in the way that I just described, but then again in the presence of an estrogen receptor peptide. Now, in the presence of the peptide, we want to find molecules that preferentially bind to 1433 when the peptide is present, suggesting that they're cooperative with the peptide. And then we'll study those in more detail. So it took a few years from 2012 when we started thinking about this until we found the right people who wanted to execute on this project. And so we had, I would say, our first sabbatical between the groups with Alina Shabesma, who was a graduate student. She was a master's student at the time in Luke Brunsveld and Christian Ottman's lab, and she came and worked with my graduate student, Ken Hollenbeck. So doing this project, they identified a number of fragments that seemed to stabilize the 1433 estrogen receptor peptide complex. And I want to show you the three ways that we tend to look at these peptide protein interactions in the presence of these compounds. So first, that primary screen is a mass spec detected screen. And we can look now, if we take one of those disulfide hits and change its concentration to a serial dilution of that compound and see how much is bound to 1433 in the absence of estrogen receptor or the presence of estrogen receptor, we can see that the fragment binds relatively poorly in the absence of estrogen receptor, and in the presence of estrogen receptor peptide, it binds much better at much lower concentrations. We can then flip the system around where we look at the peptide binding to 1433 by putting a fluorophore on one end of this peptide. And when the peptide is bound to the protein, it's seen as being much heavier, it has a slower tumbling time, and therefore we gain fluorescence polarization. So low polarization when it's not bound, high polarization when the peptide is bound to 1433. So we tune the concentration of peptide and protein so that there's a very low binding between the two, and then titrate in the compound and see an increase in that protein peptide complex being formed as a function of compound concentration. We can then do the experiment another way to measure the binding affinities of the protein-protein complex, the protein-peptide complex, in the presence and absence of the compound. So here in gray, in the absence of the compound, 1433 estrogen receptor has a KD of about 2 micromolar. Then when we add the compound at a high concentration, so a saturating concentration of compounds, say 100 micromolar, then we see a 40-fold shift in that protein-protein interaction. Okay, so what fragments did we identify? So the first fragment that we really focused on was at a non-native cysteine residue, so at this engineered cysteine residue at position 42. And this fragment is just a tiny little thing that stabilizes 40-fold. It's really a functionalized phenyl ring. The chloride binds deep into the groove, that's back here. This ether doesn't really do that much. And the rest of this just scaffolds the orientation of this phenyl ring into that pocket. And the phenyl ring acts like a little wall that creates a new binding interface, a new binding surface for this C-terminal valine residue. Okay, so it's a cute little fragment. I think that's a really nice demonstration, but what are we going to do with that? So this is now my sabbatical. So we call it sabbatical 2. I went over to the Netherlands one summer so that Alina, now a PhD student, could show me how to do X-ray crystallography. So this is the crystal structure that we solved of that fragment, same fragment that I showed you, but without the methyl ether there, in the presence of another fragment. So remember, in the fragment discovery, we can start with one fragment, find another one that binds nearby, and then link them. So how are we going to go about doing this? So Alina realized that another screen that she had done, a crystallography-based screen, identified fragments like this, amidine, that made a good hydrogen bond with estrogen, with 1433 at glutamate 14, but didn't really have any function. It's way on the edge of the protein. Here's estrogen receptor peptide. It's a miles away from a molecular point of view, but they did sit there. So we had this neutral binder and we had our stabilizer. So we wondered if we could crystallize them at the same time So now we have all four things in the same crystal structure: 1433, estrogen receptor, our stabilizing fragment, and a non-stabilizing fragment. And they are less than 3 angstroms apart. So when they're less than fewer than 3 angstroms apart, it just says, link me. So now our chemistry team comes in with a bunch of great ideas for how we can link these two fragments. This is Luke Brunsfeld, Adam Renslow, and Jeff Needs. So the first set of compounds that we made, there were several different amidine type fragments that had been found in the original screen, and we tried several of them with the fragment that we had that was stabilizing. And so here's the first compound that really showed. Now you're looking at the density. The density just fits very nicely for this compound. And if you overlay it, this is Priya Jashankar's work. If you overlay the original fragment with this now linked fragment, you can see that the stabilization piece of the molecule is in exactly the same place. Okay, so we could pretty quickly link these molecules in a way that maintained both the hydrogen bond and the stabilization factors. And then, then doing a bunch more compounds, we were able to increase the bulk of that compound and now make something that stabilized by about 25-fold. Okay? So that's our first demonstration of fragment linking, taking one covalent fragment, one non-covalent fragment, and linking them to make a purely non-covalent molecule that stabilized this protein peptide complex with high selectivity. So taking just a little detour, we also wanted to, okay, that idea worked starting from a non-native residue. What about the native cysteine residue? Because if we start with a native cysteine residue, we can make potentially covalent molecules that could be probes or drugs. So in looking at this, and here's the estrogen receptor peptide complex again, you see there's this big hole where fusacoccin fits, where our molecules could fit. But a lot of other protein complexes with 1433 have much smaller sites. For example, the transcription factor FOXO1 points a tryptophan into that pocket, and then the peptide turns out of the groove. So this is an internal sequence, phosphorylation sequence. Here's the phosphorylated residue. And here's this peptide turning out of the groove, pointing tryptophan into the groove. This is a much smaller and very different kind of binding surface. So this is where we can potentially get selectivity by looking at how the protein peptide complex leaves a space at the rim of the interface that a molecule is going to recognize. That molecule is going to be different to be complementary to this surface than it would to be complementary to this surface. Then there's a CRAF, which we'll talk about in a few minutes. This CRAF peptide crosses the entire groove, but it has high flexibility here. So maybe we can tackle this if the protein peptide complex is dynamic. And then other peptide complexes seem to go very deeply into that entire groove. And we wondered if maybe these were going to be more difficult to find fragments. So we have changes in the shape of the interface, changes in the amino acids that are pointing into that binding site, and also potential conformational adaptivity. And so Diana Kananova undertook this broad screen. And this is what some of the data look like from those primary screens. So here's the CRAF, here's estrogen receptor. Each of these 2D graphs represents 2 screens of the fragment library. We had about 1,600 fragments at the time. Each dot is a fragment. And across the x-axis, you're looking at the amount of binding by mass spectrometry in the absence of any peptide. And then on the y-axis, in the presence of a specific peptide. So the green quadrant is the stabilizing quadrant, where it binds poorly in the absence of peptide and better in the presence of peptide. And you'll see a couple of things right off the bat. One, some peptide protein complexes are more amenable to binding stabilizing fragments than others. So there are many more dots in the CRAF bin than there are in the USP8 bin. Second, there are some compounds that stabilize multiple targets, like the one shown in green. And then each screen also had some fragments that bound and stabilized only one target and none of the other targets. So we had both privileged or non-selective scaffolds and highly selective scaffolds identified for each target in the screen. So crystallizing this one compound, so this was the green compound on the last slide. The piece here, the binding fragment, you may notice, is exactly the same as the binding fragment we had from our first screen. But because we're tethered at a different cysteine residue now, that cysteine residue is further away. We have a longer linker to get to it. So the same binding element, different linker. When we crystallize that now non-selective fragment with estrogen receptor and with CRAF, we see here with estrogen receptor, the phenyl ring makes the same contacts that it made in the other fragment across with the valine. And the compound can bind again with CRAF, but now the CRAF peptide wraps around that compound, and the compound interacts with a C-terminal threonine. I mean, with a +1, so the residue next to the phosphorylated residue is a threonine. When you overlay the compound binds exactly the same way in both structures. And it stabilizes these two peptides, which then bind to the compound in different ways, one with an edge on and the other wrapping around. So Markella, very talented postdoc, said, I can make this selective. This is no problem. So we have this non-selective compound. And if we start with this molecule, this is now the same fragment, but instead of a disulfide, it has a chloroacetamide. We found that was the best covalent warhead for this fragment series. This compound starts off with a stabilization of CRAF of about 40-fold and ER alpha of about 4-fold. So it's a better stabilizer of CRAF. So she made 4 other changes to the molecule to make it selective for ER alpha. First, adding a large group here instead of the methyls, adding rings, and also changing that alcohol to a nitrogen, the ether to an amine. Improved specificity for ER alpha. It has to do with the conformation of the compound, and also this bulky group, you can think of it as an anti-binding element. It pushes CRAF out of the way. And then in the last step, optimize the linker using these spirocycles. So now the linker is very well positioned to put the stabilizing piece of the molecule in its ideal conformation and putting the covalent moiety in its ideal conformation. Okay, so making those changes, now reverse the selectivity. Now this compound only stabilizes CRAF by 4-fold, stabilizes estrogen receptor by about 120-fold, and it has a potent EC50, so it does all of this in lower than 1 micromolar. So this looks like it has the right potency, selectivity, and physical chemical properties to take into cells. So here's the same pathway diagram that I showed you before. What we want to do is increase this estrogen receptor has these 2 options, and we want to move it towards binding to 14-3-3, shown in light blue, so that we have less binding to DNA and less transcriptional activity. So if we are doing that in cells, if we're stabilizing this interaction, we have several predictions. One is that we'll be able to see, now this is a nanobread assay, so we're transfecting in the full-length proteins. We'd expect to see in cells an increase in this protein-protein complex, now not protein-peptide, but full-length protein-protein interactions when we add the compound. So here, as we titrate in the compound, we can see an increase in the amount of protein-protein complex that we form. And the EC50 for that protein-protein interaction stabilization is about 2.4, so not so far off from the biochemical potency of this compound. A structurally related compound that doesn't stabilize the interaction shows no change in this assay. Okay, so we can stabilize the protein-protein interaction. That would mean that we would also be protecting this phosphorylation from dephosphorylation. So Amira Visser, cell biologist who worked at the National Cancer Institute in the Netherlands and in Christian and Luke's labs, set out to understand the cell biology of these compounds. So first she showed that indeed, when we add our stabilizer, here's the stabilizer, here's an analog of the natural product, we can dramatically increase the amount of phosphorylation that we see at that C-terminal binding site. So in MCF7 cells, this is a highly active estrogen receptor dependent cell line. So you would expect the inhibitory regulation of estrogen receptor to be low. So most of the estrogen receptor is bound to DNA and is transcribing DNA. So the inhibitory phosphorylation level is low. If you add fusaconcan or our stabilizer complex, you see now a dramatic increase in the phosphorylation at that inhibitory site, suggesting that we're pushing through Le Chatelier's principle towards the inhibited state. If we're pushing towards the inhibited state, then we should also be reducing the amount of estrogen receptor available to mediate transcription. And if we use a luciferase transcriptional reporter assay, we have a certain amount of transcription. We have a certain amount of transcription here in the absence of any compound. If we add 85 is the compound that is structurally related to our stabilizers, but doesn't have any stabilization activity, it shows no effect. Our active compound at 1 micromolar shows a strong inhibiting effect that's very similar to ICI. This is a compound that's used, it's an ER degrader, selective estrogen receptor degrader, that's used in the clinic. Okay, so at 100 nanomolar, ICI shows a very similar effect in this assay to 1 micromolar of our stabilizer now. So ICI acts by blocking binding of estrogen. Our compounds act by blocking probably the estrogen-bound estrogen receptor away from DNA. And if we are blocking a estrogen receptor dependent cell line, removing its estrogen receptor activity, then we should see cell death. And in fact, we do. These cell lines grow pretty slowly. So we look at this with an incucyte. So the MCF7 cells are growing here in gray. And if we add our compound, then over time, we slow the growth of cells pretty similarly to what the ICI compound does. By contrast, if we take the same compounds, the same concentrations, and we look at a breast cancer cell line that is not estrogen receptor dependent, we see no change in cell growth. So this is an on-target activity of estrogen receptor. So then sabbatical 3, Christian Ottman came to our lab and said, I want to start a company. And so he and Luke Brunsfeld and I started this company called Ambagon. And while that company is going on, now this is a separate set of activities, but it did lead us to think about what other targets might be of interest in oncology that would be, would really break new ground for important kinase targets or important cancer targets. And so we turned our attention to this system, the RAF kinase system. Around 2019, 2021, all of a sudden, there were a bunch of amazing cryo-EM structures showing how CRAF was inhibited by 1433 binding, and then later how it was activated by 1433 binding. So this is a great example of how the same interaction can either be activating or inhibiting depending on where 1433 binds to the target. So 1433 shown in gray, these are cartoons made from the cryo-EM structures from these several labs. When CRAF, or these are BRAF structures, but we're assuming that CRAF behaves the same way as BRAF. The RAF structures, when they're phosphorylated near the N terminus at 259 and at the C terminus at 621, this is CRAF numbering, then the protein is in an inhibited state. You might call it an auto-inhibited state, but it's a 1433 bound inhibited state. And in this state, the domains that bind to the membrane and that bind to RAS are sequestered from that. So they're hidden by 1433, and that blocks it in an inactive state. When 251 is dephosphorylated, now RAF kinases can be released, bind to the membrane, bind to RAS, and become activated through dimerization, where a second RAF complex, RAF monomer, binds to 1433 at the C-terminal sites. So the C-terminal site is constitutive or activating, depending on your perspective, and the 259 site is inhibiting. So we wanted to stabilize the inhibitory site and see if we could make selective CRAF inhibitors through this mechanism. So Markella, again, designed a molecule. I won't go into the whole design process, but designed a very efficient little molecule, shown here in purple, that binds behind CRAF. So here's the CRAF peptide. This molecule looks like two-sided tape. So it binds to the 1433 on one face and binds to the peptide on the other face and stabilizes this interaction very dramatically. This is a 240-fold stabilization of this interaction. It has no stabilization of the constitutive site on the C-terminus. So the compound is selectively stabilizing the peptide sequence that's on the inhibitory side. And now Holly, I think Holly's picture is coming up in a minute, a graduate student who just finished her PhD, Holly Vickery, then set out to see, well, what would we see in cells? So what we would expect to see in cells, if we're stabilizing this inhibitory site, first we would expect to see, again, by nanobreadth, that we could stabilize that protein-protein interaction in cells, which we can. An inactive structurally related compound has no effect. We would stabilize the phosphorylation state, again, by sequestering, so same as estrogen receptor. An inactive compound has no effect on the phosphorylation state, but these two stabilizing compounds both increase phosphorylation at that site. And if we increase the inhibitory phosphorylation at that site, we should block binding to RAS. And so we see that these compounds cause about a 50% or so inhibition of this RAS-RAF interaction. We also see a blockade in the RAF dimerization, so CRAF dimerization, and we're still looking at the effects on phospho-ERK and transcription. But it brought up this really interesting idea that a graduate student, Johanna Verta, who's still in the lab, said, well, okay, blocking oncogenic signaling can be really challenging, but there's also a rare disease called Noonan syndrome that's caused by the same activation of the same pathway that's activated in RAS-RAF-driven cancers, but it's called Noonan syndrome, and it's a much more subtle effect. This is now a 2 to 4-fold upregulation in this pathway through mutations that lead to a developmental disorder compatible with life, but a number of defects, of developmental defects, the most important of which is cardiac myopathy. So there are several mutations in Noonan syndrome, and the one most relevant to us today is this CRAF 1433 interaction site. Type 5 Noonan syndrome have mutations in CRAF, and it won't surprise you that these mutations all map to the inhibitory binding site for 1433. So these mutants here is the 259 site. The disease mutations are right around that 259 site. So there are two different reasons that this could cause increase in MAP kinase signaling. It could inhibit the ability, these mutations could inhibit the ability to phosphorylate the site. They could also inhibit the ability of phosphorylated protein to bind to 1433. So Johanna and Holly looked at both of these, showed that the phosphorylation at 259 is in fact greatly impaired, leading to upregulation in phospho-ERK. So binding is impaired, phosphorylation is impaired rather, and phospho-ERK is activated, which is consistent with the mechanism of 1433 binding at this site. Additionally, these point mutations can weaken the binding, even of fully phosphorylated contexts, they can weaken the binding to 1433. So these are the binding affinities of phosphopeptides with these disease mutations binding to 1433. And the question was, can we use some of these molecules, like this molecule here, to bring these mutated binding sites back to a wild type affinity? And in the case of 3 out of 4 of these mutants, we could in fact bring it back to a wild type level of binding, which now we're taking into cells to show that that wild type level of binding increases the phosphorylation of these residues that are of these mutants that reduce phosphorylation. If we can increase the binding, we can increase the phosphorylation. And these are that data quantified. So we can get about a 2 to 3-fold increase in phosphorylation for these Noonan syndrome mutant proteins binding to the inhibitory site. So our next step here, showing that we can increase the inhibitory state, can we go into a cell line like a cardiac myocyte that has a Noonan mutation and see if we can correct the defects caused by that Noonan syndrome mutation, cardiac myopathy and sarcomere dysregulation. So around this time, we had sabbatical number 4, the next generation, where Holly and Johanna spent a month in the Netherlands learning crystallography from Marloes, Bente, and Amira. So just to quickly summarize what we're trying to do here, through this intercontinental collaboration, we learn a lot about each other and a lot about how science is done at these different sites, all in the service of really trying to tackle this systematic molecular glue approach. So in the case of 1433, our goal is to bind to the composite interface with these small molecules. So they gain selectivity and potency by recognizing both 1433 and the target. So we have a composite interface that creates this binding site for the molecules. And we're able to modulate undruggable targets, mutated protein-protein interactions in disease states. And one of our next projects is extending into new protein-protein interaction networks. So what did we learn about doing this molecular glue development systematically with 1433 that we can now apply to other hub protein client complexes? So again, I want to come back to the slide that I started with. This amazing team, most of them are here, but these are everybody's names. And I don't know, I was looking through this photo and I realized, oh, we have a Dutch person. So they're infiltrating the group. So Guido was visiting us for a few months to develop a separate collaboration, developing molecular glues for a completely different target class. And so it's great to have him and deepen the collaboration now into new target classes. And so with that, I think I'll just leave it there. And I'll be happy to take any questions.
Thank you so much, Michelle, for that wonderful talk. So I'll start with a question which is in the Q&A box. And if anyone has more questions, please write them in the Q&A box. All right, so the question is, in your ER story, downregulation of the cis-containing 1433 isoform would lead to resistance to the molecular glue. Is this something you tried to look at? So that's number one. And then there's another question right after it says, is the redundant nature of the 1433 isoforms a more general liability in terms of possible resistance mechanisms?
Yeah. Okay. So this is a very astute question. So if I'm working in oncology and I'm targeting a specific cysteine residue in one isoform that's not required for the biology, then that does give us a potential mechanism of resistance. And yeah, it could be a mechanism of resistance. We haven't seen that if you look at DETMAP or other genetic maps in cancer. You don't really see much in the way of mutations of 1433. Usually, if anything happens, it's silenced. Or the sigma isoform is most likely to be silenced, in fact, because it is a p53-dependent gene. So that is definitely a potential mechanism of resistance, which is why we are also looking at reducing that mechanism of resistance coming up by looking at non-oncogenic or non-oncology targets, and also looking at that fragment linking approach where we are now independent of the isoform. So those compounds should bind to every isoform.
Thank you, Michelle. There's another question here in the chat box. So with regard to the ER story, how does the presence of estrogens affect the potency or cellular effects of your molecules?
We can really see that most easily in the luciferase assay, in the reporter gene assay. So if we really remove all of the estrogen, we use charcoal stripped media, remove all of the estrogen, we see very low transcription from in the luciferase assay. Then when we titrate it back in, just picomolar is enough to get maximal transcription or to get significant transcription. So it's hard to measure. We don't see huge differences in the binding. So I think the phosphorylation is dynamic enough, maybe independent of the estrogen receptor, estrogen binding to estrogen receptor also. A little bit, I'm not sure that the picture that we drew there is very accurate. You may have noticed I didn't say that it traffics it in and out of the nucleus. What we can see is that it does or doesn't bind to DNA. But whether it's what estrogen receptor role, what estrogen roles estrogen plays in that process and dimerization and nuclear translocation are matters of debate. We haven't. Yeah.
Thanks, Michelle. More questions are coming in. So here's another one. It says, is there possibility of these compounds you mentioned to be developed for treatment of Noonan disease? It's along the lines of what I was thinking to ask you as well. So
yeah. So here's the challenge with Noonan syndrome. Well, here's what I was going to say. So before we started working in this area, I would have assumed we would have to treat patients either during development in utero or right after birth, and that's one thing that makes developmental disorders very, very challenging for drug discovery. But at least working with the now adult iPSC that are now differentiated into cardiac myocytes, those defects in the cardiac myocytes can be corrected. So with genetic changes in the cardiac myocytes, and we want to see if we can reproduce that with small molecule. So that suggests that there is some dynamics to it. Whether we can correct cardiac myopathy that's already developed in the heart, I don't know. But that would be the big challenge.
And just to follow up on that question, how about the ER stabilizers, like the 181 molecule that you saw? Are you thinking of taking that to the clinic? I mean, it looks like it was functioning at least in MCF7 cells, similar to the ICI that is in the clinic, right? So if I remember correctly, would you think that would be something you can take to the clinic and would better work for cancer treatment?
Yeah, so Amira's paper on the biology of that just went into bioRxiv recently, so people can look into that if they're interested in what other things she looked at there. So we looked at organoids and primary systems, primary cells, and the compounds do show very interesting activities in those more complex in vitro models. We haven't taken them into animals, and in fact, we haven't really looked at the PK yet to see what the exposure is going to be in tumors in animals. So I think it's worth doing. I tend to be a little conservative about these things compared to academics. Like I want to do the PK and then the toxicity and the selectivity before we go into an animal model. But these might be 1 or 2 iterations away from something that we could put into animals. Then whether you want to use it as a clinical, take it to the clinic, that's a business question as much of anything else. So demonstrating differentiation enough to put another estrogen receptor inhibitor in the clinic is challenging. So we have to take it further and further by ourselves before I think somebody would get excited about the money that it would cost to put it through the clinic. So somewhat a business question rather than a science question.
Makes sense. Thanks, Michelle. You have one more question, if that's okay. We're actually a couple of more questions. If you can hold on a few more minutes, if you have time, if that's okay.
Yeah, sure.
Right. So the question here says, more generally, since 1433s have so many clients, how do you assess the specificity of your compounds to determine whether you might be affecting the 1433 residency of untargeted proteins?
Yeah. So we can measure peptide protein complexes at scale. But what about the full-length protein-protein interactions? And if there are hundreds of them in any given cell line, then how can we be sure that we're being selective enough? Their proteomics is the best way. And Ambagon has a really great proteomics team. So they're really the experts in this. And you can pull down 1433, see what proteins come with it in the presence and absence of stabilizers. You can also look at changes in the phosphoproteome, so direct targets become more phosphorylated, and then if there are downstream targets in that pathway, you can see whether those are modulated. So I think that's the best way to get on 1433 off client selectivity. Then there's for our covalent compounds, we also want to look at what the compounds are binding to that might be not related to 1433, that might cause toxicity or confuse the biology readout. So that we do by using chemoproteomic platforms.
Thank you, Michelle. One more question here. For covalent glues, how do you imagine the risk non-specific binding to other cysteines on other targets and the potential for tox?
Yeah, that's what I was saying about chemoproteomics. Exactly. And the tools to look at those proteome-wide selectivity for small molecule, covalent small molecules is a very mature field now. So we have a number of different approaches that we could take, like the ones that have been pioneered by many people. Ben Cravath and his progeny done some beautiful work there. We also will add alkynes and use click chemistry, biotinylate the protein and pull down, see which proteins come down with a biotinylated version of our compounds. Those are the best ways to see what's present. And then you have to validate them and see if they are causing biology that's either toxic or will give you a false idea of what your molecules are doing on your target of interest.
Fantastic. Yeah. I don't see any more questions. So thank you, Michelle. This was a great talk. We appreciate you being here. Thank you everyone for attending. Hopefully you all attend our next webinar that is coming up in September. We'll announce that on our website soon. All right. Thank you, everyone. Thanks again, Michelle.
Thanks, everybody.
You all have a great one. Bye.
Bye-bye.